Genetic fingerprinting. So the principles of genetic fingerprinting. So not all organisms' genomes code for proteins. Some of the genome consists of variable number tandem repeats, or VNTRS. And these are base sequences that don't code for proteins, and they just repeat next to each other. So for example, CATG is repeated here 3 times. So it's a repeat of the non-coding base sequence CATG. And the number of times these VNTRs are repeated differs from person to person. So the length of these sequences in their nucleotides differs too. The repeated sequences occur in lots of places in the genome, and the number of times a sequence is repeated at different places in the genome can be compared between individuals, and that is called genetic fingerprinting. The probability of 2 people having the same genetic fingerprint is low because the chance of 2 individuals having the same number of VNTRs at each place they're found in the DNA is very low too. So, producing genetic fingerprints. Genetic fingerprints can be compared between individuals. So step 1, PCR is used to make DNA fragments, to make more DNA fragments. A sample of DNA is obtained from a person's blood or saliva, and then PCR is used to make many copies of the areas that contain the VNTRs. Primers are then used to bind to either side of these repeats, and so the whole repeat is amplified. Different primers are used for each position under investigation, and the product is DNA fragments where the length in nucleotides corresponds to the number of repeats the person has at each specific position. So for example, one person may have 60 nucleotides and another person may have 100. A fluorescent tag is then added to all the DNA fragments so they can be viewed under UV. Step 2 is separation of the DNA fragments by gel electrophoresis. So to separate out DNA fragments, the DNA mixture is placed into a well in a slab of gel, and it's covered in a buffer solution that conducts electricity. An electrical current is then passed through the gel, and DNA fragments are negatively charged, so they move towards the positive electrode at the far end of the gel. And shorter DNA fragments move faster and travel further through the gel, so the DNA fragments separate according to the length, and this produces a pattern of bands. Step 3 is analysis of the genetic fingerprints. So after the gel has been running long enough, the equipment is turned off and the gel is placed under UV light. Under UV light, the DNA fragments are seen as bands, and these bands make up the genetic fingerprint. You can also add a DNA ladder, which is a solution of known lengths of DNA fragments. So it's a mixture of DNA fragments of known lengths that allows you to work out the length of the other bands in the gel, which means that 2 genetic fingerprints can be compared, or you can compare the length of the sample fragments to the DNA ladder. And you could also compare 2 fingerprints. For instance, if you have some DNA from a crime scene and different suspects, you can find where the bands match. And the more matches, the more likely it is that person. Or if you wanted to use it for for paternity testing, because you get half of your genetic code from the mother and half from the father, half of the bands should match with the father. So if both fingerprints have bands at the same location on the gel, it means they have the same number of nucleotides, and so the same number of VNTRs at that place. It's a match.